Welcome back to CEC Gurukul lecture series on bioinformatics, and we have already studied several aspects of bioinformatics in earlier lectures. Like, apologies for this small gap in between, but now we carry on with that foundation where we have already studied what is bioinformatics, how do we apply this, what all makes up bioinformatics. Like, we talked about several types of databases and gene databases, sequence alignments, phylogenetics, and metagenomics as well. So today I plan to study or maybe teach you the genomics, proteomics, transcriptomics, and metabolomics. So these four themselves are very vast terms, but we try to investigate and understand the relations between them and how these four together have brought a revolution in biology, or in fact in the science in general. So let's begin with this, and as usual I start with a cartoon and a funny note to this, that searching your name in Google is also an addiction and is also linked to a character which is located on genes. And it is latest news that scientists who are working on the several aspects of genomics, proteomics, metabolomics have found this gene, that if this gene is not functioning properly, then you look at your name in Google more than the normal range. So that was on the funny note, but with this example at several places we will understand how this genomics, proteomics, and metabolomics, etc., have been useful and interesting and a revolution in the science. So I go further. I think it makes sense to first explain you these terms. What do they mean? So I already told you it's a part of revolution. There is a omics revolution, and what is this omics revolution? Is that earlier the science, or maybe genetics or metabolites, proteins, they were individualistic, meaning we used to study them gene as one entity, and we used to call that as genetics. But in this omics revolution, what has happened is the unit of study has become much vast. Rather, it's an organism, and we have holistic approach, meaning we treat the earlier, the unit of study, when it was gene, we used to call this as genetics, and when it was protein studies, but now the unit has become an individual. If the unit is genome, we call this genomics, and proteomics, and metabolomics, and transcriptomics. And I'll be taking you to these detailed meaning of genome, proteome, metabolomes, etc. But here I just want to tell you that these refers to the studies where we have holistic approach, and we talk about genome, proteome, metabolome, etc. and it's a sort of revolution. So just like to begin with this small picture, and this here, when we are biology students, we have studied the central dogma of biology, and we've been central dogma of molecular biology. And what does this say to you is that DNA is transcribed. There is a process called as transcription, wherein what happens in the transcription, we have RNA synthesized from DNA. So this process is called as transcription, and all types of RNA are synthesized by this. And then from transcription, there is another process that is called as translation, and in the translation, with the help of several enzymes and ribosomes and transfer RNAs, a polypeptide is synthesized from the amino acid as a building block, and this leads to synthesis of proteins. So synthesis of polypeptide from RNA with mRNA as a template, we call this process as a translation. And these proteins which are synthesized, they undergo several post-translational modifications, because of which we get the active form of proteins which we use. So this, what I told you, is the central dogma of molecular biology, on the basis of which we get the phenotypes. We are determined by our genes, whatever we carry in our genes, and only that phenotype is expressed. But what do we do in genomics, transcriptomics, proteomics and metabolomics? In genomics, I will not talk about DNA or a gene. I will talk about all the genes in an organism. It can be a bacteria, it can be human being, it can be monkey, it can be chimpanzee. So when I am talking about the complete DNA of one organism, we call this as genome. Complete DNA. We consider all of them together. So this is a kind of holistic approach. Same way, the transcript, that is the RNA, which is synthesized by taking instructions contained in the DNA. This transcripts, when we consider all the transcripts together, we call this as transcriptosome, all the organisms together of an organism. Then same way, proteome, when we talk about all the proteins in an organism, and metabolomes, when we talk about all the metabolites in an organism at one go, we call this as metabolomes. And I already told you, study of this as a unit, we call them as omics. And these type of studies have brought a revolution that we call as omics revolution. So not only transcriptomics, genomics are the terms, because I said it's a revolution. You see these terms written everywhere, like exomes, for everything, like this secretome, chaperones, allergome. So everywhere where they have expanded their unit of study, they have started calling this as ome, and this is a part of omics revolution. So we have all types of omes present, but here we'll be studying only 4 of these. I said these are, I told you already, genomics, proteomics, transcriptomics and metabolomics. In the first section here, I'll mainly talk about genomics and proteomics, and in the next section I take up the transcriptomics and metabolomics. So one very interesting question here, which will help you to understand what is the difference between genetics and genomics and what we have done exactly. So let us take the classical example of genetics of the Gregor Mendel when he studied pea plant. And if you remember in your earlier the school classes, in fact, you are told about the reason of the success of Mendel, why Mendel was successful. He is called as father of genetics, and he studied the 7 characters in the pea, and these characters were pea shape, round or wrinkled, pea color, green or yellow, pod shape, constricted or inflated, pod color, green or yellow, flower color, purple or white, plant size, tall or dwarf, or maybe the position of flowers was also a character. And when we used to learn the reasons for success of Mendel, I hope everybody remembers, these reasons were that he only chose 7 characters, one thing, and the other thing was that luckily he chose those characters, they were not showing incomplete dominance, or they were controlled by only one gene. So what does that mean? Because luckily he chose the system which was very, very simple, and he considered one gene at one time. There is another thing which make it very, very interesting to learn, that how we reached from genetics to genomics is that it is always written when you write this thing that why Mendel was successful. Another good reason for this was that he was a statistician. He could do the statistical analysis of all progeny, all phenotype he is getting, and those were the reason of the success of Mendel. And if I take it further, Mendel considered 7 characters, but in genomics I am considering all the characters or all the genes, maybe 20,000. When I am talking about the human genome, I am considering all 20,000 at one go, right? And my genes are not simple as Mendel had considered. Our genes are showing incomplete dominance. They are controlled by several genes at one time, and the other thing which Mendel chose at that time was statistical analysis. Of course, it was manually possible at that time because he chose only 7 characters. but here we use computers, algorithms, all bioinformatic resources. So with this small example, I tried to convince you is that the genomics would have never come into existence without the bioinformatics. So in genomics, we take into consideration all the genomes, all the genes, of an organism. I said if it is human being, we'll talk about all 20,000 genes here. And the other thing is we extensively use computational resources, and I also said our genes are very, very complicated, interrelated, and it involves lots of other things which we'll talk about after we go further. So again, why I took all these four together, all these four topics and titles together, is that they are interrelated. I told you the central dogma of molecular biology, because genomics, the genes express themselves as transcripts, and the transcripts is the blueprint for protein synthesized, and the metabolites which are accumulated in any cell at any state of physiology is controlled by the proteins, meaning they interact with the proteins, and proteins use them to catalyze many reactions in the body. So we took all four together because it is understood that all of them are interrelated, and presence of one and another makes a difference, and the purpose of all these is again to apply them for human welfare, especially in crop production, dealing with the environmental problems, or maybe the diseases, medicine, and further applications for humans. So we start with today's lecture, and this was just an introduction. I was just trying to tell you that what all is the foundation or the basic things which we must have known before we can come to the exact objectives of today's talk. And here we have four types of omics, I told you already, which we'll be talking about one by one: genomics, proteomics, metabolomics, and transcriptomics. And we'll talk about the bioinformatics tools which are applied in here, and applications and outcomes of these. And because we're considering all four of them together, so respectively the time allotted to them will vary. So starting with the genomics, is the study of genomes. And what is a genome? If you want to know the definition, is the complete set of genes in a cell or in an organism. All the cells of an organism have the same copy of genes. So if you say cell or an organism is the same thing. So we talk about the complete set of genes. Say if we talk about, let's take the example of human genomes. So when I'm talking about a genome with humans as an example, I would say I will talk about all 23 chromosomes at one go, understanding the human behavior or the genetic control or gene expression, and not only the 23 chromosomes which are located in the nuclear DNA, I will also consider the DNA present in the mitochondria. So in genome, we consider the complete genome that includes the chromosomes in the nuclear DNA and the DNA present in the mitochondria. So in genomics, we talk about the genome, and I gave you the definition with the help of information from human genome. And these genomes, they are very, very complicated. As I said, human genome or maybe chimpanzee genome, but they can be very, very simple also, where we have only more than 400 genes present, that is methanococcus, where this genome is very small in size. But it is interesting to know here is the first genome which was sequenced was the East chromosome, and once it was done, then it was tried to sequence more organism, and then a bacterium Haemophilus influenza was sequenced. And when few organisms were sequenced, of course in the beginning when this work started, it took much longer time and the techniques were much slow, and they were very expensive. So that is why only small organisms could be tried, and it made sense to try small organisms or prokaryotes because they offer us the simplified systems, meaning they don't have any introns and complicated regulatory elements present in them. So studying them makes sense, but there were several few genomes at that time. In 1990s those were sequenced, and then came the era of 2003 where we had human genome sequenced, which has 33 billion base pairs, and we have, at that time we knew that we have 30,000 genes, but when the research is going on, now this number of genes have come to 20,000. Now we have 20,000 genes. And another thing which happened in 2007 was that personal human genome could also be synthesized. What does that mean? What do you mean by personal human genome? So there was one human genome, first human genome was sequenced, and then the first genome which was sequenced, or the personal human genome, was of that Watson, James Watson, the famous molecular biology, Watson and Crick, known for his Watson and Crick model. His genome was sequenced, and lots of interpretation considering his behavior could be made, and that was a revolution in the medicine and other things, because after a while the genome of his sister was also sequenced. So when we had these two genomes, the things were much easier. And after that there was a exponential increase in the number of genomes sequenced. There was a huge number of genomes sequenced, and this has been rising every time. What was the reason for this? I told you already, is the cheap and easy, quick methods of sequencing, like this automated sequencer which came in 1987. It revolutionized, and there was a 43-fold increase in the genomics, in the genomes being sequenced. And then the revolution again which came was because of next generation sequencing, which made it very quick and very low cost affair. So after that this genomic sequencing was in practice, and it was very easy, and it has come in the excess of every human being. In fact, I remember when the first genome was sequenced, it used to take a very long time, maybe in years, and the cost was in $1 billion. And then after that it came down several times, and the cost was reduced to a cost which is less than the cost of a Mercedes car, and now it is very not that expensive that you can't think of sequencing a genome. So if I like to add, what is happening now is that we have in 2021, if we talk about in animals, this is 3278 unique animal genomes have been sequenced, meaning their nuclear genome have been sequenced. This 3278 is a huge number to have the data of these many genomes. This is available on NCBI, and this refers to 0.2% of animal species. If I talk about plants, we have, because this figure keeps changing, we have more than 800 genomes sequenced, and if I talk of fungi, we have about more than 100 genomes sequenced. So when we have this much of data available with us, it requires complex algorithms, lot of bioinformatics people who analyze this data, take out meaning out of these, and I hope you could understand the need for the bioinformaticians and the information of the molecular biology which can be used for human welfare. I go further, and I am just telling you few more vital stats about human genome, taking the example of human genome. That human genome, I said, have what? 20,000, about 20,000 genes, and its size of the genome is 3.2 gigabytes, and if you say the nucleotides, it is 32,000 million base pairs. And if we see the coding sequences, out of this only we have 1 to 3% sequences which are coding sequence. What does that mean? 1 to 3% meaning of all the DNA which we know in humans existing, most of it is not expressed as proteins, as a phenotype. Rather, it is non-coding. They do not carry any phenotype. Rather, they have a role in the gene regulation or in the gene expression or control or other type of gene or the system in the human beings, or not only human beings, in the other organisms. So these refer to the non-coding, ribosomal RNA, transfer RNA, ribozymes, small nuclear RNA, and several types of regulatory RNAs. It also includes promoters and the other gene regulatory elements, and several of the DNA which they are structurally, they have important structures and replicatory roles like scaffolding regions, telomeres, centromeres in the chromosomes. Those also needs to be sequenced, and there is a lot of DNA which does not carry any gene. That DNA is also popularly called as junk DNA. So recently the female human genome was sequenced, and without Y chromosome it was said to contain 19,800 sequences, and the coding sequences here are about 1.5%. So we have huge number of line, sign, introns in these sequences. That makes these complicated. It makes sense, here I cannot, but still I cannot go further without telling you these three terms, that the genomics is of three types. One is functional genomics, then we have comparative genomics, and then we have structural genomics. What are these three things? The functional genomics is where we assign a gene a function, meaning what phenotype it carries. It is controlling the skin color or hair color, or maybe it is controlling the cancer or the cell division. That we call as functional genomics. Then we have comparative genomics, where we compare the genome of one organism with another, and then we know how we are similar to each other, and this leads to the making of the phylogenetic evolution. Also we can study this way, and we can know what has evolved from which one. And when we talked about the phylogenetics in earlier lectures, maybe you can know that the comparative genomics has a huge role. Then structural genomics also exist, and there we have this 3D structure. We try to identify the 3D structures. So I hope you believe this could not be done without bioinformatics, and we needed to have bioinformatic resources here. So again, I talked about genes and genomes, and we'll talk about proteomics, transcriptomics, and metabolomics in the next section. Thank you very much.